Hi everyone, this is Mrs. Bustamante. In this video, we're going to talk about prokaryotic gene regulation. So remember, basically we're talking about bacteria cells, and we're going to talk kind of how bacteria cells work to help make proteins that our body needs in order to digest things, or a protein that we maybe need to make in order to form some other function. And these bacteria cells, where are they? They're probably in our gut, in our intestine, right? Where we either want to break things down or make things up, but they could work like this wherever they are, right? So how do we regulate them? With two ways, really: regulatory sequences and regulatory protons working kind of in conjunction with each other. And there's quite a bit of vocabulary in this that you really need to take some time to make sure you can understand and explain. One region we need to know about is something called an operon. Okay, and when we draw prokaryotic genes, we typically draw them like this. It looks kind of like a line. We're going to have our DNA, and we're going to have this region that we call an operon. Okay, our operon is just a group of genes that work together to regulate and perform some sort of common function. They're either going to make something or they're going to make something to break some stuff down. They have some role and they all work together. Now, there's kind of a bonus to one thing being activated, or a group of genes being activated all at one time, is it's a little bit more efficient, right? We also are going to have some regulatory sequences with regulatory proteins, and those sequences will code for that protein typically, and that helps us control what happens, we say typically downstream in that operon region, those genes that work together. So when we look at our operon here, it kind of includes our promoter, our operator, which we'll talk about in a second, and then these things called structural genes. And our structural genes are what we think about when we transcribe to go on and make proteins, right? So these are the parts that are going to code for our proteins to make that mRNA, and then that mRNA, right, can be translated into proteins that fold and go on and do other things, right? We're talking about this in bacteria cells today, so prokaryotic cells, right? And we said it's in our gut, so it's used to break down things or adjust to what we eat, right? And we need to be able to do that pretty quickly and efficiently and all the time, right? Are we trying to look at sugars or proteins or amino acids? What's some of the other vocabulary? Well, we talked about our operon, a group of genes that works together, right? Well, we're going to have a promoter there. It's in green here. There's a reason. I like to think promoter means go, okay? It's our regulatory sequence. It's typically non-coding. I mean, it doesn't code for a protein, and it really just serves as our binding site for RNA polymerase. It's kind of like the go here sign, start here sign, promoting it, if you will, to go on. Then we have our operator. I put our operator in red because it's going to correspond to a color here in a second. It's really our regulatory sequence. It's also non-coding, doesn't code for a protein, but it's going to serve as a binding site for our repressor protein. So basically the protein that's going to say stop or go based on its location, and that can change a little bit, right? So it's a type of regulatory proteins. Think about operators as controlling what's happening. It's kind of our control of what is happening, or a way that we do control this gene expression in prokaryotic cells. And then I have structural genes. You'll notice it's in rainbow colors. Why? Because they can be lots of different things, right? And there can be lots of genes grouped together, so lots of different colors, okay? These are your coding genes. These are the parts that are going to go on and make proteins, or the functioning part of our operon. They're going to code for different enzymes typically. Enzymes are proteins, right? And those have a job, okay? So we can control lots of genes at the same time through this operon function. And if we remember progs, that's kind of a way for us to remember the order. We go promoter, repressor, operator gene, and then structural genes typically. So there are two types of operons we're going to talk about. We're going to talk about inducible operons and repressible operons. First, we're going to talk about inducible operons. They're usually catabolic, meaning they're going to try and digest something. Their job is to break something down. So we say that their default is off. Why is their default off? Well, they only want to break stuff down if it is present. So it's typically off. Now we say it's inducible because to induce means to be able to change something to on, right? So we can turn it on by a molecule that we call an inducer. Turn it on, right? And we say this is positive regulation. The most common example of an inducible operon is the lac operon, okay? It's a gene in bacteria that's job is really to be able to break down parts of lactose, right? The sugars of lactose in the body. Okay, so let's take a moment and talk about this. Here is my DNA molecule. Okay, we say this is upstream. This is my 5' end. This is my 3' end of my DNA. And let's use some things that we know. Okay, we have our regulatory gene or our regulatory area, right? We have our promoter here, and we have a regulatory protein that's going to be coded for here. Notice it is upstream of my operon area where I have my promoter, my operator, and then my genes. Now, what do these genes mean? I labeled them Z, Y, and A. Why? There could be more of them. There could be less of them. They're just a way to label what they are, and they're going to go ahead and produce some mRNA that we will need to go ahead and turn into a protein, right? Okay, so typically what will happen? Okay, typically we have RNA polymerase. Just like we saw before, RNA polymerase will bind at my promoter region. Remember, RNA polymerase's job is to add RNA molecules that are complementary to the DNA molecule there that code for a protein. Okay, so this RNA polymerase is going to go along my DNA and it's going to hit my regulatory protein. In the lac operon situation, when I hit this regulatory protein, that regulatory protein is going to make something called my repressor. Okay, now my repressor protein is going to fit over here at my operator site. Okay, and it's important that it fits over here at my operator site. Now RNA polymerase will go along and it can bind to this promoter site as well. But because my repressor has bound to my operator here, RNA polymerase cannot go along and transcribe these genes downstream of it. It's like blocked. It's like a wall. Okay? And this is what happens in a bacteria cell when lactose is not present. Okay? And if we think about this, the reason this makes sense is if we don't have lactose present and our lac operon genes over here, their job is to break down lactose. We don't want to produce the enzymes to break down lactose when there's no lactose, okay? So let's say you eat something, now all of a sudden there's lactose present, okay? When lactose is present, it produces this, it's actually like, let's just say lactose, we say induces this fit, okay? It actually kind of produces an enzyme that we call the allolactose, which will bind to my repressor, okay? When lactose is present, I produce allolactose, which is my inducer, okay? It's going to bind to my repressor. And when allolactose binds to my repressor, I have a conformational change, and it unbinds from my operator. Okay, now it is no longer blocking my operator, so DNA polymerase can go ahead and transcribe the rest of my gene. Okay, it's gonna make that mRNA copy of genes Z, A, and Y, right, and continue over and over and over again. Why is that important? So I'm making the enzymes that will go ahead and digest lactose. Now eventually though, right, lactose will be used up. There will be no more lactose. What will happen? RNA polymerase bind to the promoter here. My regulatory protein will make my repressor, and because no lactose is present, my repressor will bind to my operator again, and RNA polymerase will no longer transcribe those genes to make the enzymes that digest lactose. Okay? That's your lac operon. You should be able to explain that and do that pretty fluidly and easily. So in summary, right, inducible are usually catabolic, trying to digest something. Their default is off, and we have to induce them or stimulate them to turn on by some molecule, typically an inducer of some kind, right? So we say this is positive regulation. The more we produce of that inducer, the more we transcribe the gene. And the lac operon's job really is to break down lactose, which makes sense here. All right, so our other type of operon are repressors. Our example of this is going to be our trip operon. Unlike inducible, they are typically anabolic. They're making something to build something up. So their default is typically on, but they can be repressed, or think about turned off in a way. They're going to be repressed by something called a co-repressor. Things that are going to work together. Think co-workers work together. They're going to work together to turn it off. And in our example here, our trip operon's job is going to be to synthesize or to produce tryptophan. Okay, so same idea, DNA 5', 3', regulator gene area. We have a promoter. It's going to make some sort of regulatory protein. We have our operon area with a promoter, an operator, our genes. And here's going to be upstream. First thing, our normal is on. So what does that mean? That means RNA polymerase typically can bind my promoter, right? And it can go ahead and transcribe these genes. What do these genes do when we're looking at tryptophan, right? These genes, when we're looking at tryptophan, actually are going to make enzymes that make tryptophan. So that's our typical on, if you will. All right, so most of the time, RNA polymerase is going to be making tryptophan, right? Now, however, as tryptophan is produced, what's going to happen? Well, eventually, my RNA polymerase bind to my promoter, make my regulatory protein, okay? Now, here's my repressor protein. Here, I have my repressor protein, but you'll notice under these conditions, it doesn't fit at my operator, right? It doesn't have the correct binding that I need in order for that to work. So in order for this to work, I need to have what we call is a co-repressor. And my co-repressor will be made when I produce a lot of tryptophan. Here's my co-repressor, and they bond together. These bond together, my repressor and my co-repressor, that's basically my tryptophan. What happens? Well, there's a magical shape change. And now my repressor and my co-repressor can bind to my operator site. So now what happens? Well, now when I reach my promoter, right, it's like hitting this wall. I can no longer go ahead and transcribe my structural genes down here, meaning I'm no longer making tryptophan, right? My repressor and my co-repressor bind to the operator, and there is no more transcription of tryptophan. Now, eventually, right, if tryptophan gets low, this will release, right? And we can go ahead and do DNA polymerase, right? When I get enough, my DNA polymerase come over here, make my regulatory protein. Again, it will combine with my inducer from tryptophan, shape change, and do it all over again, right? So what I learned, repressors like our trip operon, right, usually anabolic, build something up. Their default is on, but we can repress them with a co-repressor that will turn them off. And the trip operon's job is going to be to synthesize or to produce tryptophan. Now, what should you be able to do? Use those, explain those, but really those concepts of inducible and repressors are applicable to anything with a name. And you should be able to have one explained to you or shown to you and be able to talk about, is it an inducible or repressible example, and explain it or tell how you know. Hope that was helpful. Have a great day.